Hey guys, we're here to talk about translation, the second half of the protein synthesis process. So we left off in the last session where we had modified the messenger RNA, dealt with the introns and exons, and the messenger RNA left the nucleus and went out into the cytoplasm and headed to ribosomes. So in translation, we are synthesizing a polypeptide, so amino acid chain, using information from the mRNA. So we really are changing from the language of nucleic acids to the language of proteins, two of our different macromolecules that we started off the year with. This process occurs at the ribosome, so all types of cells will do this. And the nucleotide sequence becomes an amino acid sequence. That's I think of translation as going from one language to another, and here we're going from the language nucleotides to the language amino acids. tRNA, that's this little structure, the blue structure right here, is a key player in translating mRNA to an amino acid sequence. And so in reality, you can see where it kind of gets its name from this T shape that it has. Okay. So when we're talking about our tRNA, our transfer RNA, it has what we call an anticodon. Remember the little sets of 3 on the messenger RNA, we called those codons because they had the code on them. So the anticodon, you'll notice, complements the mRNA. But also notice that just like with our DNA strands, remember, we're always dealing with antiparallels. So the 5', 3' are going to be opposite each other. tRNA will also carry the amino acid that it codes for. So ACU codes for threonine. And then the enzyme aminolysyl tRNA synthetase, we know it's an enzyme because of the ACE, is responsible for attaching the amino acids to the tRNA. So there's an enzyme that's holding on to the threonine onto this tRNA. And so when it has that amino acid, we call it charged. Basically what we're saying, it is ready for this process. We've got all the pieces put together and we're ready to go. That's what we mean when we say it's charged. Okay, so see anticodon, and here's our codon. Notice they complement each other. And they code for a specific amino acid. So in the ribosome, this is where the actual translation will occur. And so you'll notice that the ribosome has 2 subunits, a large one and a small one. And the mRNA, you'll notice this purple strip is between the 2. The prokaryote and eukaryotic ribosomal subunits will differ in size, so they're not going to be exactly the same, but they both have ribosomes. In prokaryotes, the small subunit is about 30S and the large one is 40S, whereas in eukaryotes, the small one's about 40S and the large one's about 60S. Okay. The large subunit is going to have 3 specific sites within it. You notice we have an A, we have P, and we have E. So the A site is what we call the amino acid site. Okay. And it's going to hold the next tRNA that's coming in. So this new one is going to come land in the A site. It's kind of like a prep site. We're getting everything lined up and ready to go. P is the polypeptide site, because this is where we're actually building that polypeptide or amino acid chain. And E is the exit site, so as it moves into the E site, then it will release and leave. So, how does tRNA interact with mRNA? Through the anticodon. Remember, the anticodon complements the codon. What codes for the amino acid? And the answer is the mRNA codon. This is the biggest place that I see people mess up on this process, is they're trying to use the tRNA to actually say what amino acid it is when it's the mRNA that has the code on it. So the mRNA tells us what it is. tRNA just carries it and helps match it up. What are the 3 sites on the large ribosomal subunit? So that's our A, that's like our arriving. Our P is basically where we're building, we're making the polypeptide chain, and E where we're exiting. Okay, so translation has 3 stages, just like we did with transcription. So we have initiation, elongation, and termination. Gee, those should sound familiar. So we're going to start with initiation. Translation begins when the small ribosomal subunit, down here at the bottom, binds to the mRNA. See how we've attached to the mRNA. And a charged tRNA, notice it has the amino acid attached, so it's charged, binds to the start codon. Start codon is always AUG, and so the anticodon would be UAC. AUG, which stands for methionine, is the start codon. It will always start with AUG on the mRNA. So this guy is going to bind with that guy. tRNA carries methionine, which is part of what we have with start codon. And then once this binds on there, the large subunit, this large piece, will actually bind on top. So the first tRNA with the methionine will always go to the P site, and every other tRNA will come into the A site first. But we've got to get that initial one in there to bind on the large ribosomal subunit and get the process started, initiate it. Then we will elongate. So the next tRNA comes into the A site. mRNA is going to move through the ribosome, and the codons are going to be red, and as we shift, that's how we keep bringing in new tRNAs. As each mRNA codon codes for a specific amino acid, and we do use a codon chart to determine what it is. And remember, we always put the mRNA into the codon chart because mRNA has the code on it. Since all organisms use the same genetic code, it really supports that evolutionary concept of common ancestry, because obviously they all have something in common, so there must be something that connects them. And so we continue with elongation. The codon recognition, so we recognize the appropriate anticodon pairs to the codon in the A site. And then we scooch over, the peptide bond is formed. And then translocation, the tRNA that's in the A site moves to the P site. The one on the P site goes to the E site. And then the A site is now open for the next one, and the E site's going to release, exit. Okay, and then we get to termination. Termination occurs when a stop codon in the mRNA reaches the A site. So when you look at the chart, you'll notice that some of them specifically say stop. And a stop codon doesn't code for an amino acid. It literally means stop. It is a signal for a release factor that kind of breaks or hydrolyzes the bond that holds that polypeptide to the P site so that we can release the polypeptide, because the whole point is we're building this polypeptide to send it off where it needs to go. And then all the pieces kind of disassemble, break apart again. So I want you to think really quickly, talk to each other about the 3 stages of translation. Okay, so before we move to protein folding, we're going to take a minute to review primary, secondary, tertiary, quaternary protein structures. Okay, so remember, primary is the chain of amino acids. So a line of people hand to hand connected. Secondary is either going to coil or fold, so think slinky or a folded fan, due to the hydrogen bonds. So we took that chain of people and every other person takes a step forward while still holding hands. Tertiary, we're going to start having side chain interactions. This is what I always think of as the popularity situation. You know how some people, there are some people everybody wants to be around, and there are some people that nobody wants to be around. Well, the same thing happens based on these side chains, where we'll have certain side chains that attract to each other, whereas other side chains tend to repel each other. And when we're attracting or repelling, this is causing the chain to twist and coil and fold. And then quaternary, not all of them will do quaternary, but some do. And we have 2 or more polypeptide chains that will actually interact with each other and fold together. So, protein folding. Because remember, one of the things we learned about proteins was shape determines function. Shape determines function. If it doesn't fold or shape correctly, it won't work correctly. So as translation is taking place, as we're elongating that chain, that polypeptide chain is actually starting to interact with all of the pieces and is starting to coil and fold as it's being built. The genes will determine that primary structure. Remember, we've got adenine, then we have threonine, and then we have tyrosine, and then we have a lysine, and then we have a serine. It's just the series of what amino acids set next to the next one. And that will ultimately be what determines the final shape, because each of these will interact in a specific way. Some polypeptides will actually have to have what they call chaperone proteins to make sure they fold correctly, that they're kind of watching the process and they're nudging, hey, you need to fold here, or we have to change this a little bit before you can fold correctly. And so sometimes you do have a few extra little protein molecules that have to be present to make sure it folds correctly so it can function correctly, because shape determines function. So I have a couple of other YouTube videos, and I've dropped the links onto Schoology, but I want you to kind of watch, that helps put the whole process that we've been talking about in these last few videos together. And the last thing I want to touch on is this concept of retroviruses, because this is a slight variation to the normal process that we would think of. So retroviruses are things like HIV. Actually, influenza is a retrovirus as well. COVID is a retrovirus. So they don't work the exact same way that what we've just talked about, because first, the information has to flow from RNA to DNA. Retroviruses actually have DNA as their key genetic or hereditary material. And so when we inject the RNA, we also have something called reverse transcriptase. And so basically, we're having to turn the RNA into DNA, which then can become part of the cell genome to be processed. So we're going to go from RNA to DNA, and we use reverse transcriptase to do this because we're doing this transcription process backwards. We're reversing it. And so this is how we take viral RNA and turn it into DNA, which then can be used to make messenger RNA and go through the process. Okay, practice-free response. All living organisms need a transfer of information to allow for genes to direct the synthesis of proteins. Explain how genes are transcribed and translated to produce a functional protein. And then another, compare and contrast translation in prokaryotes and eukaryotes.